Here we go with the third and final set of notes in our cell division series. This one is on meiosis and how we produce our sex cells, our sperm, our eggs, or better known as our gametes. As you probably recall, meiosis is another type of cell division. This one produces 4 haploid cells instead of 2 diploid cells like mitosis. It happens mostly in your sex cells. This is how you're going to be creating your sperm and your eggs. And basically, we're not going to get into all the steps of meiosis, but it's pretty much the same as doing mitosis twice. And the really important thing about this is that it increases the genetic diversity of an organism in 3 different ways. The first way it creates genetic diversity is through a process called crossing over. And this is where homologous chromatids swap pieces. And basically what happens is during metaphase of meiosis, the chromosomes line up in the middle just like they do in mitosis, but in the meiosis' version of metaphase, they line up next to each other, so they probably look something like this. So I drew my 2 different chromosomes as different colors. They're lining up next to each other, and sometimes they overlap a little bit. They're going to line up next to their homologous pair, so these guys are both basically the same chromosome, one version from mom, one version from dad, and they overlap a little bit. And what happens is they can overlap, and then when they break apart, they actually swap a little bit of DNA, so that after they pull apart, they might actually look more like this. Now you've ended up with 2 chromosomes that are slightly different from the way they started. This guy is mostly the red, but it has a little bit of blue, and this guy is mostly the blue chromosome, has a little bit of the red. So whatever messages were on these little bits have now swapped pieces. So that's one way to increase genetic diversity, crossing over. The next way we increase genetic diversity is through something called independent assortment. And basically what this means is that chromosomes from the mom or chromosomes from the dad separate randomly. So the daughter cells are a random mix of mom and dad's chromosomes. So that might look, using our little red and blue chromosomes, something like this. So you can see it's not just all the reds on one side and all the blues on one side. It can be a random mix. And this is just how I randomly drew it. It could have been the blue one over here and the red one over here. It could have been all blues and all reds. It just depends on what happens. And as we continue through meiosis, you're going to end up then with a cell that looks like this. These chromosomes have been pulled apart, and eventually this is going to turn into a new cell, and eventually this is going to turn into 2 different sperm or eggs. And so as you can see, the different sperm or eggs are going to be different. So depending on which sperm is actually successful in fertilizing which egg, you could get very, very different combinations of babies. A little bit of interesting math as far as this is concerned. It's really the way you figure out the number of possibilities of different ways you can get different babies is basically 2 raised to the number of chromosomes we have, or number of pairs, which is 2 to the 23rd. And so that comes out to be more than 8 million possible combinations of gametes. And then you throw in all the different combinations that you could get with crossing over, and that increases that number dramatically. So this is going to create babies that are very, very, very different from the adults, which is the whole point. The third way that meiosis leads to more genetic diversity is basically just through sexual reproduction. Because remember, the whole point is to make daughter cells that are haploid, so again, only have half of the number of chromosomes. So in males, you'd be making 4 different sperm. In females, it's a little bit different. You make 4 eggs, but as you see here, like this egg is bigger than this one, and then over here, this one is even bigger than this one. So basically, the body is putting all of its eggs, no pun intended, into one basket. So it's putting all of its energy into making one supercell. So you end up with one cell that survives in 3 what are called polar bodies that basically just deteriorate and fade away. So you end up with 1 egg or 4 different sperm. And then through sexual reproduction, you're going to be taking half of your genome and combining it with half of another person's genome, and so you're getting some more genetic diversity in that way. And then the last thing we want to talk about during meiosis, something that can happen called non-disjunction. And basically, this means junction is a coming together, disjunction is a separation. We have a non disjunctions. We have a non-separation. So non-disjunction is when chromosomes do not separate during meiosis when they're supposed to, and this leads to what's called trisomy, or a missing chromosome. If you're missing a chromosome, that almost always leads to death. So if you look at this during meiosis 1, you can see that these two chromosomes separated from each other like you're supposed to, but these guys, they didn't separate. They stayed together. So you look at this side here, and these guys, instead of having two sets of chromosomes, they only have one. These guys have three, and when you get to the gametes in the end, this guy only has one chromosome. It's supposed to have two. This guy's only supposed to have two chromosomes. It has three. These guys have extras. This guy is missing one. Or if it were to happen later on during meiosis 2, that second stage, again, they should have separated apart, right? So this is what it should look like. You should get some normal cells with two chromosomes. Instead, you're getting one with one chromosome, one with three. So if you had to have non-disjunction happen, you'd want to have it happen in your second meiosis, because then you still get a couple cells that are normal. If it happens really, really early, that's a mistake way, way early in the process, and that's always bad. You're either going to get trisomy, or you're going to be missing some chromosomes. And so some of the more common ones, trisomy 21 you've probably heard of, so when you end up with an extra 21st chromosome, this is Down syndrome. Some of the symptoms or the signs of Down syndrome usually have some physical abnormalities, the slanted eyes, sometimes folds over the skin of their eyes, usually it's mental retardation, and often there's other effects as well, like their heart or other internal organs don't work as well. The most common trisomy is actually trisomy 16. This one happens the most, but it's always fatal, so you almost never see it. Whenever you do have a chromosomal abnormality, this is usually really bad. Most times when you have extra chromosomes or you're missing a chromosome, you're not even going to be born. That would result in a miscarriage. The one kind of exception to this is if you're missing one of your sex chromosomes. So if you're missing a Y chromosome, and so instead of being XY, you're just X, that is something that is survivable. And you probably only have infertility issues. But pretty much everything else, if you have an extra chromosome or if you're missing a chromosome, you're either going to not survive or you're going to have some very serious birth defects. And with that, we are done with our cell division unit.